Statements may include statements regarding, among other things, the efficacy, safety, and intended utilization of our product candidates, our future R&D plans, including our anticipated conduct and timing of preclinical and clinical studies, our enrollment in patients in our clinical studies, our plans to present or report additional data, our plans regarding regulatory filings, potential regulatory developments involving our product candidates, potential regulatory inspections, revenue and reimbursement estimates, projected terms of definitive agreements, manufacturing capabilities, potential milestone payments, our financial position, and our possible uses of existing cash and investment
Brief CAPR Therapeutics announces positive top-line results from pivotal Phase 3 HOPE-3 study
that are subject to change and involve a number of risks and uncertainties that may cause actual results to differ materially from those contained in the forward-looking statements. These and other risks are described in our periodic filings made with the Securities and Exchange Commission, including our quarterly and annual reports. You are cautioned not to place undue reliance on these forward-looking statements, and we disclaim any obligation to update such statements. With that, I'll turn the call over to Linda Marbán, CEO.
Good morning, everyone. This call this morning represents the culmination of 20 years of work. This technology was originally conceptualized on a poster that was handwritten in Capri, Italy, more than 20 years ago. And through all the years of understanding the mechanism of action, trying to find the perfect way to deploy the powers of what was once CAP-1002 and is now deramiocel, we are now proud to be able to present today the data that was released in a press release this morning that showed that the HOPE-3 Phase 3 pivotal clinical trial of deramiocel to treat Duchenne muscular dystrophy has shown statistically significant improvement in both skeletal and cardiomyopathy. Today, in order to update you not only on the data from the trial, but also talk about our future plans and then to contextualize the relevance of this data for the Duchenne community, I am joined by my esteemed colleagues, A.J. Bergman, who you just heard from, Dr. Michael Binks, our Chief Medical Officer, Mr. Mark Awadala, our Chief Development Officer, Drs. Kati Mahari and Nathan Hogan, our statisticians here at CAPR, Dr. Craig McDonald, a physician that needs no introduction in the Duchenne community because of his prominence in the clinical development, understanding of, and treatment of Duchenne muscular dystrophy and other neuromuscular disorders, and Dr. Jonathan Soslow, by far considered the leader in the world of understanding Duchenne cardiomyopathy and has published extensively on the natural history of this disease process in order to contextualize this important data for you. Next slide, please, AJ. You can see in this very simple agenda that I'm going to be walking through some of the very basics of the trial design, how we did it, how we conducted it, the rigor by which this data was collected, the fact that we have now an adequate and well-controlled study which has been asked for by the FDA, and I will give you all of those details, and we look forward to your questions at the end. Mostly what I'd like to say before I begin this call is my email, my phone, my text messages this morning has exploded in the last few minutes since the release of the press release by the most important people in this whole experimental design that we've built, and that is those with Duchenne muscular dystrophy and their families. These boys and young men and their families have hope. This is the first clinical trial that I am aware of where there is statistical significance, clinical significance, and also anecdotal support for the fact that deramiocel changes the life course of these boys and young men. Next slide, please, AJ. I think most of you on this call know about Duchenne muscular dystrophy. It is an X-linked disease. It means primarily expressed in boys and young men, although there is a growing body of evidence that the mothers of these boys, as well as the sisters of these boys, have manifestations of the disease as well, and we look forward to be able to be providing therapeutics for them in the future. Deramiocel is the largest protein in the human body. It acts as a cushion and a glue, so it protects cells from damage, and it also helps it holds its structure. The pathogenesis of Duchenne muscular dystrophy is the worst of the muscular dystrophies because those with this disease do not have any dystrophin that is naturally made by their body. Therefore, all of their cells are always undergoing decline, ultimately breakdown, and early cell death, leading to the loss of progressive muscle groups. Duchenne takes typically between 25 and 30 years to take the life of a boy or a young man, and every day during that progression of the disease, they are losing some aspect of their ability to function. We hope to be able to attenuate the course of this disease with deramiocel moving forward. I'd also like to highlight that one of the most important aspects of deramiocel and sequelae of Duchenne muscular dystrophy is the inflammation. Inflammation is caused by the constant breakdown of muscle. The immune system is put on alert that there is something going on that is bad, and as the immune system is activated, it leads to greater cellular breakdown. So if you can find a therapeutic where you can attenuate inflammation and fibrosis, you have a potential success, and I think that is what we have seen here today. Next slide, please. Deramiocel has been under clinical development for over 10 years in the treatment of Duchenne muscular dystrophy. We started with the HOPE-Duchenne study that was published in the Journal of Neurology, where we showed a reduction in the amount of scar in the heart and trends toward improvements in cardiac function. We then showed that we could multi-dose, switching to an intravenous paradigm in the HOPE-Duchenne open-label extension, which rapidly led to the conduction of the HOPE-2 and the HOPE-2 OLE clinical studies. The HOPE-2 and the HOPE-2 OLE clinical studies have shown not only that we can reduce the amount of damage done to the muscle, that we improve cardiac function, that we improve skeletal muscle function, that we are addressing an unmet need that nobody else has been able to address, and that is the primarily non-ambulant patient population, and that we see long-term, multiple years, 4 years coming into 5 years of safety as tolerability as well as efficacy. As you know, we filed a biologics license application based on the HOPE-2 open-label extension data, as well as the natural history data collected by Dr. Jonathan Soslow and published in Circulation Heart Failure. And what we are talking about today is the data that was asked for by the Food and Drug Administration to supplement that BLA in order to prove the fact that deramiocel works in treating Duchenne muscular dystrophy. What's most exciting to me and what I want to highlight on this relatively busy slide, the culmination of this work is HOPE-3, the data that we have put out this morning and we will continue to discuss now and moving forward. It has been predicted by each one of our clinical studies. In HOPE-Duchenne, we showed a reduction in cardiac scars measured by LGE. You'll hear in the next few minutes that that was substantiated in this randomized double-blind placebo-controlled trial. The improvement in upper limb function shown in every one of our clinical trials continues to be supported and now shows statistical significance in a 106-patient randomized double-blind placebo-controlled trial, and the improvement in cardiac function is again substantiated from the HOPE-2 and the HOPE-2 OLE study. Now, how does deramiocel work? Let me remind you of that. Shown on the next slide is the multimodal mechanism of action of deramiocel. We've recently published this work in a peer-reviewed journal. The measurement of the mechanism of action has been ongoing in our lab and others over the past 20 years, but what has been most important in the development of deramiocel as a drug product is the development of a potency assay that is directly related to the biologic mechanism of action of deramiocel. And that's shown in this relatively simple graph, which shows that we have antifibrotic activity, immunomodulatory activity, reminding you that inflammation is equally damaging to patients' muscles with Duchenne muscular dystrophy and other neuromuscular diseases, by the way, as well as anti-inflammatory itself, just slowing down that inflammatory response. So allowing inflammation to be reduced, allowing healing to begin, and then ultimately preventing the development of scar, which leads to the loss of muscle function. All of this has been validated, approved by the Food and Drug Administration as the potency assay moving forward, which allows lot-to-lot consistency, allows us to scale up our manufacturing, allowed us to pass our pre-licensing inspection for our San Diego manufacturing facility, and with this data, we believe we are prepared to launch deramiocel to treat Duchenne muscular dystrophy. New slide. Let me talk to you for a minute about HOPE-3, our pivotal phase 3 trial. This trial has been conducted at 20 sites across the United States. It is a randomized, double-blind, placebo-controlled trial of 106 patients. The primary efficacy endpoint is the performance of the upper limb version 2.0. The key secondary endpoint is left ventricular ejection fraction, and we have other type 1 error-controlled secondary endpoints in the mid-level performance of the upper limb. That is the ability to move essentially your elbow or your arm, an analogous task by which could be improved by preserving mid-level function is eating, brushing your hair, drinking water, hugging your mom, as Pat Furlong would say. A global statistical test, which is a feels, function, and survives type of endpoint. It combines looking at the relationship between the performance of the upper limb, left ventricular ejection fraction, and a patient-reported outcome called the PGI. We have late gadolinium enhancement. That is a dye that is used to examine the heart using MRI and allows you to determine the amount of scar in the heart. And we see that as well statistically significantly, and I'll talk about that in the next few minutes. As I mentioned, it was a 1-to-1 randomization. Patients received 150 million cells via intravenous infusion 4 times a year, and as you'll hear in the next few minutes, it was safe and well tolerated. Next slide, please. Inclusion criteria were primarily based on performance of the upper limb criteria. That means that patients had to have upper limb function with a pull entry score between 2 and 6, primarily between 2 and 5, where we took in several of those patients that were still in late ambulation, at risk of losing ambulation and still maintaining a significant amount of upper limb function. The purpose of this was to provide the opportunity for those that are at the edge of losing ambulation to get deramiocel and also potentially for label opportunities. What you can see shown here in this slide, which can be also elaborated on by Dr. Craig McDonald, who, as I mentioned, has joined me today, the ability to feed yourself or lift your arms, raise your arms above your head, are all the criteria by which patients are randomized into this trial, and we are then able to measure their functional development as a result of the performance of the upper limb, which is a 21-item scale. Next slide, please. So shown here is the demographics of the study. HOPE-3, as I mentioned, was 106 patients, randomized, double-blind, placebo-controlled. The mean age was, in the placebo group, slightly younger, 14.6 years. In the deramiocel group, 15.4 years. The overall in the trial, the average was 15 years of age. We had a slight difference in left ventricular ejection fraction at baseline, with the placebo group being slightly healthier with an entry ejection fraction of 59% and a deramiocel entry left ventricular ejection fraction of 55%. A total of 91 patients had left ventricular ejection fraction measured, and you can see that the average baseline criteria for ejection fraction was 57%. There was no difference in baseline in the performance of the upper limb in the placebo versus the deramiocel group of any measurable difference, with the overall pull entry score for those with entry scores of 2 to 3 at 48, or 4, 5, and 6 pull entry scores, an overall entry score of 58. All considered well within the standard expectations of this particular patient population. Most of our patients were non-ambulatory. Let me highlight at this moment the incredible relevance of this therapy in the Duchenne community. There is nothing that is approved actually for this patient group, and we are the one therapeutic that can be given across the life course of Duchenne muscular dystrophy to attenuate the progression of the disease. Many of the boys and young men receiving deramiocel are in their early 20s and feel and function better than they ever thought they could at this point in their life. Next slide are the safety results of the HOPE-3 clinical study. The safety of deramiocel is one of the aspects of this clinical development program that we are most proud of, and that is because, of course, the one thing you want to be able to do in drug development is provide a therapeutic that not only works, but doesn't impact the quantity or quality of life of a patient. The reason our patients in our deramiocel clinical trials, HOPE-2, HOPE-2 open label extension, HOPE-3 and HOPE-3 open label extension, have such low dropouts, even with many of our patients staying on for years in the open label extension program, is because of the safety. Primary safety concerns, if you can call them that, are extremely simple. They're mild flu-like symptoms reported in some of the patients. 25 to 35% of the patients have some version of a pyrexia, which is fever, cough. Some get itchy. A significant proportion get headache. Physicians manage this easily with antihistamines and Tylenol, and as is evident from the lack of serious adverse events, as well as the reliability by which patients come back for new treatment, is evidence of the safety as well as tolerability profile of deramiocel. This is something we stand behind. Years of data supported. Data Safety and Monitoring Board has been following our patients for years, and the FDA has received all safety data in timely filings. So we continue to be proud of the safety impacts of deramiocel. So now let's get into what you guys really came to hear today, which is the efficacy of deramiocel, because we've been talking about some of these other features for quite some time. So shown here in this bar chart is a pictorial representation of our primary efficacy endpoint. That is the performance of the upper limb 2.0 at month 12. Shown with the green arrow is actually the difference of means, which is a 54% slowing of disease, a 1.2-point change on an absolute scale. And what we can see here is that statistical significance, the likelihood that this is due to chance, is less than 3% at the p-value of 0.029. The difference is shown on the y-axis as percent change from baseline, although, as I mentioned, the absolute change was 1.2 points. Both can be represented. This happens to be the way that the statistical analysis plan was drafted, submitted to the Food and Drug Administration. We received no comments on the statistical analysis plan, and patients as well as regulators as well as statisticians find this a very easy way to quantify performance of the upper limb. And let me just explain that to you so you understand the importance of percent change. If I have a PUL score of 20 and I lose 2 points, and my colleague Mark has a PUL score of 40 and he loses 2 points, that is a very different amount of functional loss for me versus Mark. So this percent change allows us to actually quantify the loss on a per patient basis and its relevance. So again, this was something that was submitted to the Food and Drug Administration, was decided by us and multiple statisticians that we have worked with over the course of the last year, as the best way to represent functional improvements, changes, declines in this patient population. And our stats team is happy to take questions on that should you have any. In the next slide, we show our secondary endpoint, which is left ventricular ejection fraction. Shown here is a 91% slowing of disease, again measured by the green arrow showing the difference of means, and the percent change from baseline. We are using a rank change with a p-value of 0.41. 0.041. Those zeros are mighty important right now. This data is also important, a 91% slowing of disease. We saw stabilization across the board in patients treated with deramiocel, and we saw a decline in those that were placebo treated. I want to point out, and this is something I've been thinking a lot about, and I think our KOLs can provide context here. The disease Duchenne muscular dystrophy is one that takes somewhere between 25 to 30 years to take the life of a boy or young man with Duchenne. As I said at the beginning, it is a slow and steady decline towards that inevitable and terrible outcome, the primary cause of which is loss of cardiac function. In order to do a cross-sectional analysis of looking at these guys for 1 year and to see this kind of data is unprecedented. If you can delay the progression of this disease by 91% in terms of preservation of heart function, where 65 of the 83 documented patients with cardiomyopathy at baseline have stabilization, we have a tremendous opportunity to potentially extend not only the quality of life of these boys and young men, but potentially the quantity of life for these boys and young men. And I wanted to highlight that here, the importance of treating this aspect of the disease. Next slide, please. Shown here is a forest plot. A forest plot is a special type of way of representing data. It is a way of showing that data either goes positively, which is to the right of the zero, showing that the treatment is having an impact in one way or another, and to the left of the zero would be showing favoring placebo in one way or another. What is shown in this very beautiful plot is the fact that we not only hit the primary efficacy endpoint of the PUL 2.0, the total score, we hit the key secondary endpoint of left ventricular ejection fraction with a p-value of 0.04, as I just mentioned on the previous slide. We hit the all type 1 error controlled secondary endpoints. What does that mean? What it means is that sometimes in a clinical trial, when you sort of laundry basket a lot of secondary endpoints, you can kind of lose statistical value by this concept called multiplicity. When you type 1 error control a secondary endpoint, you're controlling for multiplicity. You're saying, yes, we really do think that there is statistical validity here, and we're going to test it using traditional means. So the 3 secondary endpoints that you see here are the mid-level dimension of the performance of the upper limb, statistically significant improvement with a p-value of 0.008, the total global statistical test, the one that I mentioned a few moments ago, that measures feels, functions, and survives, is a secondary endpoint with the t-statistic, a p-value of 0.01. And then finally, one that was added as part of the cohort B of the clinical trial at the request of our cardiology colleagues is late gadolinium enhancement. Gadolinium is a dye that is used in MRI. You've probably seen it if you've heard me speak before when we talk about HOPE-Duchenne, where you see the tissue differentially stained with healthy tissue stained black and scar tissue stained white. People who are experts in reading these types of images can then quantify the number of segments of the heart that is impacted by scarring, or other ways of looking at it is the percent of the heart that is affected by scarring, and it's a very efficient means of looking at the damage in the hearts. In all heart disease, this is an important measure, but in Duchenne muscular dystrophy, it is extraordinarily important, but because what Dr. Soslow, Dr. Chet Villa, Dr. Larry Markham, many of these physicians that have studied this very different cardiomyopathy for the course of their careers has shown that this fibrofatty replacement is a slow and insidious killer of these boys and young men. They develop scars slowly. They don't know what's happening. Many of them are off their feet. Their hearts are not working as hard as they could if they were playing basketball or soccer. So they're kind of oblivious. And then ultimately there's so much scar that the heart actually can no longer compensate. They develop a fulminant heart failure, and ultimately nothing that clinicians can do can pull them back from the edge. So the fact that we see a reduction, and a statistically significant reduction in the amount of scar with a p-value of 0.02 is probably one of the most amazing aspects of this trial. We are attacking this disease at where it is causing the most potential damage, and we are looking forward to following the boys and young men that are prescribed deramiocel to see that they have extension not only of length of life, but quality of life, but also length of life. Next slide, please. So many of you who have heard me speak before have seen this particular slide. I like it. I call it the good player in the sandbox slide. Deramiocel can be used with any therapeutics that are currently approved, currently available, or under clinical development that we know of, whether it's an exon skipper or a gene therapy that is trying to attack the disease at its source, which is the lack of dystrophin, or some other types of antifibrotic or corticosteroids. In fact, we required every patient in every trial to be on standard doses of corticosteroids because we believe that that is the standard of care that has been most efficient at extending life in these boys and young men. Deramiocel goes well with all of them. There are no negative impacts, and we actually believe that we're going to see synergies over time in this type of polypharmacy, because if you can reduce inflammation and you can reduce fibrosis and you can drive repair, and then you have these other therapeutics that are in there that could be adding dystrophin or repairing the gene, we then have an opportunity to really attack the disease at both sides of its mechanism of destruction, both the lack of dystrophin and inflammation and fibrosis. Next slide, please. So where are we going from here? This is obviously probably the most important day in the history of CAPR and the history and the development of deramiocel. The name of the company, as I mentioned, comes from the little island on Italy in which we first discovered the seminal work that has led to this point. We have met the phase 3 primary efficacy endpoint of the PUL 2.0. The key secondary endpoint of left ventricular ejection fraction, both achieving statistical significance of p equals 0.03 and 0.04, respectively. As I said earlier, statistical significance was achieved in all type 1 error-controlled secondary endpoints. Deramiocel is a first-in-class therapy designed to treat not only Duchenne cardiomyopathy, but also the skeletal muscle myopathy. And we made those seminal discoveries now multiple years ago in our preclinical studies, and now it has been validated in multiple clinical studies, culminating in what I consider to be probably one of the very best clinical trials that's been done in the understanding of Duchenne muscular dystrophy, randomized, double-blind, placebo-controlled, 106 patients, highly overpowered at greater than 90%. We really have measured all of the bells and whistles here and done a stellar job of taking this to this end point. Safety and tolerability, one of the most important aspects of treating any type of disease, because of course you want your patients to feel and function better, with consistent clinical experience supporting the safety and tolerability of deramiocel. Obviously, the whole world knows that we received a complete response letter. We thought we were on track to PDUFA last year based on the HOPE-2 open label extension data, as well as comparing the cardiac data to Dr. Jonathan Soslow's cardiomyopathy natural history study. The FDA had felt that that was adequate data for a label. The administration, upon review of the data, decided that there was not adequate data. They issued a CRL. We have now answered their concerns by offering them data that is randomized, double-blind, placebo-controlled, an adequate and well-controlled clinical study. We're going to submit this data in response to the CRL. We are hoping to get a relatively rapid PDUFA date right now or up until our Type A meeting in August. They were telling us it would be a Class 2 resubmission, which is about 6 months, so look for sort of the conservative estimate of a PDUFA in July. But because we now believe that this data substantiates all of their concerns, we've passed pre-licensing inspection, our commercial manufacturing facility is ready to go. Cohort B, which is the aspect of the trial that was done was done using drug made at our San Diego commercial facility, shows independent statistical significance. We are ready to go. And our patients, their families, and all of the people in the Duchenne world that have been following this therapeutic for years, I think today we should raise a glass and celebrate that perhaps we are first time providing an opportunity for these boys and young men, especially those that are non-ambulant with no other options in front of them, to have an opportunity to extend their lives. And with that, I will again offer my thanks. I'm joined here by my colleagues who can answer specific medical, scientific, or statistical questions, and I look forward to continuing to work with you as we take this towards launch and commercialization. Thank you.
Operator, I think you can open the line for questions, please.
Thank you. Ladies and gentlemen, we will now begin the question and answer session. To ask a question, you may press star followed by the number 1 on your telephone keypad. To withdraw your question, please press star followed by the number 2. With that, our first question comes from Leland Gershell with Oppenheimer. Please go ahead.
Good morning, Linda and team, and my congratulations on these very strong data from HOPE-3. Wanted to ask, and I know you had discussed this a bit during the prepared remarks, Linda, but with respect to the primary and secondary endpoints, percent slowing versus I think what was on clinicaltrials.gov of mean change. If you could just walk us through maybe a bit further in your discussions with FDA and perhaps part of your SAP, how that will be handled versus mean change, and if there may be any differences or further discussion that's needed with respect to the agency regarding those as registrational. Thanks.
Yeah, thanks, Leland. So, obviously there's been a lot of discussion, a lot of planning that went into the statistical analysis plan. We worked with multiple outside statisticians, James Signorovich of the analysis group, who's done a lot of work in Duchenne, and then with the patients and advocacy groups, PPMD, and some of the groups that also have worked on patient-reported outcome measures, clinical relevant outcome measures. And the determination of using percent change as a way of measuring the change using the performance of the upper limb, I personally think was one of the greatest steps that CAPR was able to make. So we even worked with ADAM International, the group that developed the performance of the upper limb, and presented the idea to them before we wrote it in our SAP. They also like it. The pull is still a relatively new measure. We're still understanding it. And as I said in my remarks, a pull score of 20 and a guy who loses 2 points on that has a very different outlook on those 2 points lost than a guy who starts with 40 points and loses 2 points. So percent change allows you on a patient-by-patient basis to assess the loss for that particular patient. And for more clarity on that, Anna Mayhew, actually, again, one of the developers of the performance of the upper limb, just published a really lovely paper that was made available, peer-reviewed in September, discussing this exact issue, and that's actually where a lot of these ideas came from. In terms of absolute change, we saw that as well. So the FDA has said that the clinically meaningful change in the performance of the upper limb would be 1 point. We see 1.2 points, many of that change attributed to mid-level pull, which is their arm, which is also validated in video assessments that we actually have in front of us. We just can't show today. So look forward to the future of being able to see on video what is actually happening to the arm function in these guys that are on deramiocel. The good news is we see statistical significance in the absolute change as well. It's a p-value of 0.05, so just making the cutoff, but that's all you've got to do. So whichever way the agency wants to look at it, we hit it, and we look forward to having that conversation with them. We really don't think this is going to be an issue that's going to roadblock us at all.
Thanks. Just great. And one follow-up. As the results that were presented were across the entire HOPE-3 population, just wanted to ask with respect to cohorts, B versus A, obviously B from San Diego, which is the facility that's been, fully inspected and passed and all that, versus A from LA. Just wanted to hear of any concerns or any possible items that may come up with respect to approvability of deramiocel, given the data set across both A and B. Thanks.
Yeah, so, we were delighted. We had built a statistical analysis plan which allowed us to look at A plus B and then drive that primary towards cohort B should we not achieve significance with A plus B. We hit the primary at A plus B, so that's great. But B did even better than A. We think that's largely because we put really stringent quality assurance, quality control measures in place. Our potency assay was approved and now is done in every lot. And so every batch of drug that comes out of San Diego goes through a much more rigorous type of evaluation than from our Los Angeles facility, just because we were developing it for commercial use. So, the p-value for cohort B was really quite extraordinary, and this will do several things in our minds. Number one, it shows the efficacy of deramiocel produced by our San Diego facility. Number two, and perhaps even more importantly, it shows that an efficacious product can be made by a manufacturing expansion, so this allows us to have significant opportunity for expanding our manufacturing capabilities and treating any and all of those worldwide that ultimately can benefit from deramiocel, whether with Duchenne or other indications. And the other issue, of course, is that it really closes a lot of holes, we believe, that FDA could raise any concerns about manufacturing or CMC because of the efficacy of this product, as well as us passing PLI already in preparation for approval. So I think this is one of the best findings of the study, is how well cohort B did, and I'm very proud of the data from this aspect of the trial.
Thanks very much, and again, my congratulations.
Oh, thanks, Leland. You've been great to work with.
And the next question comes from Ted Tenthoff with Piper Sandler. Please go ahead.
Great. Thank you very much on this glorious day. Congratulations. I know how much hard work's gone into this and really how meaningful this is going to be to boys who suffer from this terrible disease. I wanted to get a sense for the response to the BLA. What really goes into that, to the reply, to the CRL? Is it just the submission of this new data set? Are there any other components or anything else that needs to be included in that submission? And you mentioned the potential to accelerate the review. Maybe tell us a little bit about that strategy. Thanks.
Yeah, so in response to the CRL, we are going to be submitting this data. We submit all of the data, and we will address all of the issues that were directly raised. So obviously everybody's seen it because the administration put it out, but we would be happy to share. And so they addressed concerns regarding whether we had an adequate cardiomyopathy patient population for a cardiac label. We do. So we have 65 out of 83 of our patients, as I mentioned, were diagnosed with cardiomyopathy at baseline. 92% of the patients in the trial were on cardiac meds at baseline. That was another concern raised in the CRL. So we feel really great about our cardiac opportunity, the LGE, and we have very good indication that we're going to be able to answer all of their concerns regarding the clinical data with this very stellar data set. We had already answered the CMC, the chemistry, manufacturing, controls issues that had been raised in the CRL, so we're very confident in that. Because we've had some time to think and our teams are never quiet, we're also going to be submitting additional mechanism of action data where we will be able to support with further analyses the actual mechanism of action which we've been talking about. I showed a very simple slide. There's hundreds of pages of scientific work that has gone into understanding how this works.
Excellent.
So, the final aspect of your question, Ted, which is acceleration of the PDUFA date. We didn't think that the CRL was issued fairly originally, but we understood it. Okay, it was a small data set. There were a lot of questions. None of the questions raised in the CRL were ones that we hadn't heard from the agency before. We were glad that we had the opportunity to file the BLA under the previous administration. We were glad that they could see through the small trial and some of its warts and bruises to the potential efficacy. But now with this absolute manifestation of efficacy, hitting statistical clinical significance, doing a clinical trial that is how a clinical trial should theoretically and practically be done, we're going to be asking the agency for a rapid review, and we will hopefully use some of the political capital as well as the families and advocacy groups to help us work with FDA to get that accelerated.
Great, excellent. Well, congratulations and best on these next steps.
Thank you so much, Ted, and thank you for standing by me for all these years.
My pleasure.
Thank you. And the next question comes from Joe Pantginis with H.C. Wainwright. Please go ahead.
Hey everybody, good morning, and congratulations as well. This is really fantastic after the path you followed, building this case here. I know it's been difficult, but it's very rewarding now. So two questions to start. First, can you give a little perspective with regard to the LVEF percentage increases and how this reflects clinically to the patients? And then number two, you just spent a bunch of time describing, the responses to the CRL components. What would you consider, if any, the worst or the most demanding case that might be still outstanding? Thanks a lot.
Yeah, so I'll take the last part of that first. I don't think we have any. I think that this addresses every single one of them. Like I said, I felt like if we didn't have this data set coming, I would have fought to the end to get a BLA approved based on the data that we had. I believed in that data. It's been published in the Lancet, published in the Journal of Neurology. Patients have long-term, over 4 years of efficacy. You can't turn your head to that. So I believe strongly in what we had. It's not even icing on the cake. It's like, the wedding cake. Like, this is it. This is the best that you possibly can have. And so we don't feel like there are any issues. In terms of clinical relevance, One of the greatest pleasures of my career has been getting to know Dr. John Soslow, who is a caring physician but also an incredible academician. John, I hate to put you on the spot, but could you possibly tell everybody a little bit about the clinical significance of this data, what it means to you, to your patients, and sort of to the field?
Yeah, absolutely. No, thank you for asking me. So, as they mentioned, the 2.4% difference in LVEF, that's striking because we see pretty much in study after study that there's a 2 to 3% decline in LVEF in these patients once they develop cardiomyopathy. And we and others have shown that LVEF is a very strong predictor of mortality. And in fact, it's shown that a 3% decrease has a hazard ratio of over 1.3 for association with mortality in those patients. So they have a 32% increased chance of having a mortality event for every time their LVEF drops by 3%. So this is clinically extremely meaningful for these patients. And what I'll say is that we treat these patients with cardiac meds that are adopted from the adult literature. There are no approved cardiac medications. It's an unrelenting progressive disease. And honestly, those adult meds really don't work very well. So we and others have honestly grown tired of going to these patients' funerals. Like, we're not making enough of a difference with the drugs we have. And so a drug that can slow or stop cardiomyopathy progression is really a game changer in this field.
That's fantastic. Thank you very much for that, and looking forward to a potential positive FDA outcome. I think you have the data to show that, so congratulations again.
Thank you, and thank you, John. That even brought tears to my eyes. Thank you. Questions?
The next question comes from Kristen Kloska with Cantor. Please go ahead.
Hi, good morning, everybody. Let me also add my congratulations, and you should be proud of all the young men and boys that you've been able to help with this therapy. So I had a few questions. Just first on the SAP change, I understand the rationale, especially as patients come in at different baselines, but can you just help contextualize for us? Did you bring this idea to the FDA in the meetings prior to the database lock, and then did they echo that they were supportive of it? Just trying to understand that, because I think there's a lot of focus on that this morning.
So explain baseline differences? Is that what you're asking, Kristen? I didn't understand the question. I'm sorry.
No, I'm saying I understand the rationale for why the percent of disease slowing makes more sense, but if you can just help us understand when this change happened in terms of your FDA dialogue, and again, just reinforce that they were supportive of using that.
Yeah, thank you so much. Sorry, for whatever reason, the webcast is a little funky for everybody, including us today. Yeah, so as I said, this was sort of iterative thinking that developed with us, with our statistician colleagues, with talking to the ATOM group that developed the performance of the upper limb. Anna Mayhew's paper in September, as I mentioned, kind of blew the field wide open as to, what's really important and how do you quantify and qualify progression, as well as working with some of the advocacy families in learning what really matters to them and how to measure their function, either decline or stabilization. We've submitted the percent change opportunity to the FDA as part of our statistic analysis plan. It's now been several months since we submitted it, but we definitely long ago passed any 30-day review period. We have, put it in all of our documents. It was in our final SAP that was submitted prior to database lock. So we don't expect any concerns from that. But in sort of the beautiful belt and suspenders way, we also see statistical significance in the performance of the upper limb in terms of absolute change, which is, going to be, whichever way you want to look at this, it's a win. We won, according to Hamilton, the musical. We don't really anticipate that there's going to be problems in dealing with whether absolute or percent change. I'll use this moment to sort of talk about the heterogeneity of Duchenne muscular dystrophy, and I think there's kind of a misnomer in genetic diseases, especially in diseases where there's a mutation that affects one protein. It should be so easy to fix, and I think that's sort of the kind of the Kool-Aid we all drink when we go into this as our careers, is that this got to be easy to fix. But what you realize is that anything that happens in the human body is very complicated. And so the fact that we can see statistically, which is obviously mathematically, how likely is this data due to chance, improvements in this patient population using this measure of functional performance and cardiac improvement is really quite game-changing, both for the world of drug development, but also for the families and the boys themselves that are impacted.
If you met the endpoint on the mean absolute change as well. I think that's important this morning. And then I know you had previously talked about that the agency would be willing to exercise regulatory flexibility. I think the way Wall Street initially interpreted that is if you missed on PUL and maybe hit on LVEF, there would be a chance there. But obviously in this scenario, you hit on both. So I'm curious how you're thinking about what the actual addressable patient population will be. Cardiomyopathy is about 80%, but then since you hit on the skeletal endpoints, one can argue that supports essentially everybody. So I'm curious how you're now thinking about the patients that would be eligible for this therapy.
Yeah, so obviously we now have changed our optics a little bit, achieving both statistical significance as well as clinical significance in the skeletal muscle myopathy. As we had been instructed by FDA to study over the past 10 years, we always considered ourselves a therapeutic for cardiac, and so the skeletal muscle was something that they felt that they could measure and therefore decide efficacy on, so we have that. We also have improvement in cardiac function, long-term stabilization. As I said, even in this one-year study, a 91% slowing of disease, and Dr. Soslow and his colleagues are working tirelessly to try and find therapeutics, and now they finally will have something in their toolbox should we achieve approval with which to offer these boys and young men. The most important thing, in getting to the heart of your question, no pun intended, was who are we going to try and market this to? What's the label going to say? We have over 40% of the patients in a correlation analysis that was done had improvements in both cardiac and skeletal muscle function, with over 70% of the patients having a response in either cardiac or skeletal. So we're going to go back in and respond to CRL to keep our cardiomyopathy indication open. We don't want to submit a supplemental or new BLA or anything like that. And then when we have our labeling discussions and we talk about the data, we will certainly ask also for a label for the skeletal muscle myopathy based on the strength of this data. All of this will be determined in our meetings with the FDA after we present this data to them. But even, almost anybody, even a high school student, can look at this data, look at the CRL, and think that there's a wonderful opportunity for approval here.
Thank you so much. And if I may ask the physicians on the line, I would love to hear how they're potentially considering using this in their practice now that they've seen this data. Thank you so much, everyone.
So, Linda, do you want me to take that? This is Craig.
Yes, please, Craig, if you could answer that question. And then maybe John could provide.
Yeah, so Craig McDonald, I have been national PI on the HOPE-3 trial and have been involved in the HOPE-2 trial as well as the HOPE-2 OLE. So I have probably, the greatest long-term experience using Deramiocel clinically in an open-label context. So in terms of really being able to see patients who I know are on therapy. And so I think the question really pertains to who I would want to use this therapy in. I think it's really quite reassuring from my perspective that to see really the consistency of results across the placebo-controlled trials of HOPE-2 and HOPE-3 and the long-term HOPE-2 open-label extension. And I find that to be really compelling. But as far as who I would want to use this therapy in, I think, first of all, the trial itself was conducted in patients that were 10 years of age. There were late ambulatory patients who had 10-meter walk-run times greater than 10 seconds. We know those patients 100% of those patients will lose ambulation over the next 24 months. So we refer to those patients as sort of approaching loss of ambulation or the late ambulatory population. And in addition, a large portion of the patients were non-ambulatory patients that had at least hand-to-mouth function, PUL entry scores of 2 or greater. But I think it's important to keep in mind that patients who entered the open-label extension have also had pull entry scores of 1, and there's been demonstration of relative stabilization of the pull, even in those more severely affected patients. So I would say, first of all, I would want to treat non-ambulatory patients, any patient with any deficit of 1 point or more on their total pull score. I think that would be kind of at the ceiling range. And I think in terms of the floor value, I think if you've got a pull entry score of